Biologist Simone Haupt has come to Haifa for a project that holds great promise for the future. She's working at one of Israel's leading institutes of technology, Technion. Haupt's research is in stem cells. Together with an Israeli colleague, Michael Amit, the German scientist is trying to develop a standard procedure for making human stem cells. They want high volume and consistent quality. If the team is successful, it would be a big step towards healing diseases like Parkinson's. Stem cells are undifferentiated cells that can be turned into most types of tissue. But that ability makes working with them in the lab a challenge.
The biggest problem is keeping them from differentiating, meaning forming human cells, for example, for the heart or brain or whatever. It's about finding just the right cultivating conditions, so that the cells remain what are called pluripotent cells, which can be made into just about anything.
Over a decade ago, the researchers from Haifa, working together with scientists from the US, explored stem cell potential in work with embryonic stem cells. These were harvested from embryos left over following successful fertility treatments, for example. A few days after fertilization, embryonic stem cells can be removed from an early-stage embryo to generate more stem cells. But this process destroys the embryo. Many Christians view this as the destruction of potential life. But Judaism provides clear guidelines.
According to our religion, life is not considered as a potential, or potential is not considered as a life. You need definite issue, implantation of the embryo, developing of the fetus, and so on and so forth.
Haifa is located on the slopes of Mount Carmel. Jews, Muslims, Christians and followers of the Baha'i faith peacefully coexist here. Some say the diversity in Haifa reflects Israeli society's tolerance of stem cell research, regardless of whether someone is liberal or conservative in their faith. In Germany, there are much stricter limits on working with human embryonic stem cells.
German researchers must, of course, rely on cooperating with foreign partners, because otherwise we would not have access to stem cell lines at all, because we can't generate them in Germany. That's why we place a lot of emphasis on working with foreign partners.
An alternative could be another type of stem cell, one that's less ethically controversial. These are called induced pluripotent stem cells, or iPS cells. To create them, differentiated cells, for example from the skin, are encouraged to return to an undifferentiated state. Then, like embryonic stem cells, they can develop into any type of cell. The German-Israeli team has set its sights high. The researchers are working with both embryonic stem and iPS cells. Encouraging stem cells to multiply, keeping them healthy while preventing differentiation, is a question of the right cultivation techniques. One challenge is finding the right nutrient medium.
It took us about 6 months to find the right medium. And then came the hard work, which took us something like a year or so, to develop a splitting method, to improve the system, to assure that it fits with these spinner flasks, and so on.
The method uses what is called suspension technology, which employs tools like this bioreactor. Here, huge numbers of stem cells are dividing under strictly defined conditions. It's a great leap forward for the German-Israeli project, which is slated to last for at least 2 more years.
The aim of the project is to create an industrial platform that employs precisely the technology we've learned to use here. We'll combine the suspension technology with our know-how on differentiating nerve tissue cells.
Because at Simone's Institute in Germany, researchers want to use pluripotent cells to make large quantities of nerve cells. If they can do that, they will be able to test drugs for treating diseases of the central nervous system. The leap from fundamental research to a real-world application is huge, but Simona Haupt and the other scientists in the team are well on their way.